Hi, my name is Alexandru Dumitrescu, and I will present TSignal, a transformer model for signal peptide prediction. First, I will present a short biological background. Signal peptides are short amino acid sequences that form at the N-terminus of newly synthesized proteins. They are usually about 16 to 30 amino acids in length, and their role is to facilitate the translocation of mature proteins. After the translocation occurs, signal peptides are cleaved from the secreted protein by specialized enzymes called signal peptidases. I will now go through the motivation of this project, the project scope, and its formulation. Motivating our work, inefficient translocation caused by signal peptides may result in certain proteins, such as hormones or antibodies, to be secreted in lesser amounts, and this can result in diseases. Signal peptide identification is therefore of biological and medical relevance. Then, the proposed problem is non-trivial, as signal peptides can be highly variable and lack clear consensus motives. For this reason, a plethora of machine learning methods have been previously developed for this task, but none of them solve the protein identification completely. The problem becomes even more challenging as even single amino acid mutations can impair the signal peptide functionality. So for the scope of our project, we aim to devise a new fully data-driven method for signal peptide identification and cleavage site prediction. We formulate the problem as follows. Given the input, a sequence of amino acids of length n, we predict for each residue an associated label. Here, important label examples are S, which stands for amino acid belonging to signal peptide, and O for amino acid belonging to a mature protein and is extracellular. After predicting all sequence labels, we can then determine the cleavage site position as the first non-signal peptide prediction after a contiguous sequence of S predictions. The picture also shows a few general properties of signal peptides, which are usually organized into three subregions. N is the region which mainly contains basic amino acids, followed by a hydrophobic region containing mostly hydrophobic amino acids, and ending with the C region, which contains the cleavage site. In this picture, we show an overview of the considered signal peptide types. We classify signal peptides into multiple categories based on the translocation channel to which they direct the proteins. Here, we have the secretory channel, or SEC, and then the twin arginine translocated channel. And we also classify those based on the enzyme that cleaves those signal peptides. And here we have signal peptidase 1, 2, or 4. In the right figure, based on the translocation pathway and signal peptidase that cleaves the proteins, we consider 5 unique types of signal peptides and a negative class. The various signal peptide types that we consider in this work have certain properties. In signal peptidase 2 cleaved proteins, we highlight how signal peptides are always followed by the cysteine amino acid. Then, in twin arginine translocated proteins, the signal peptides directing those proteins always have a somewhat conserved twin arginine motif. I will now move on to describe our problem's implementation and the model architecture. We implement the proposed prediction task using a neural machine translation setting. Our model consists of a transformer encoder-decoder similar to the original formulation of this architecture presented in the manuscript Attention is All You Need. We train our model in a fully data-driven manner where we do not use any additional information other than the amino acid sequence and their labels. So we do not use such information neither during training nor inference. Most of our model's parameters are contained in the encoder, as we use a large protein language model called ProdBERT. having 30 layers and a hidden dimension of 1024. This model was pre-trained on 216 million protein sequences, and we transfer and further fine-tune its parameters in our model. A key advantage of this formulation is highlighted by the prediction formulation. Each residue's corresponding label at every position I will be predicted based on all previous labels and all input residues, unlike, for example, a sequence tagging approach. On the flip side, this means that the necessary one-to-one mapping between an input residue and its corresponding label at position I needs to be learned by the model. To solve this, we concatenate the sign-based positional encoding to both the output and input vector representations and note considerable improvements when doing so. Large language models are known to produce highly contextualized representations, which is generally quite useful for many predictive tasks. This, however, is not necessarily the case in this specific setting, as the exact cleavage site location should be determined by the precise location of the amino acids. Very large improvements have therefore been gained after tuning ProdBERT, and we claim that optimal contextualization amount might have been the reason. To further help the model find an optimal contextualization amount, we also concatenate one-hot representations of the amino acids to the label representations, right before the prediction layer. I will now give a few details about the dataset we used and previous methods we compare against. For our training and test data, we used the cross-validated splits gathered by the authors of Signal P6, containing about 4,000 signal peptides and 15,000 non-signal peptides. Notably, within the 15,000 non-signal peptides, transmembrane domain proteins are also translocated, but contrary to signal peptides, the transmembrane domain is not cleaved after the translocation occurs. The data is organized into 3 homology splits, each containing about a third of the data and having the maximum intersequence identity of 30%. Therefore, we train and test on highly diverse sequences. We also additionally test on 4 novel eukaryotic sequences which have unusual properties as they lack clear signal peptide subregion delimitations. Now, to move on to the previous methods, most, if not all previous methods employed sequence tagging approaches and used hidden Markov models or conditional random fields. Further, to our knowledge, all previous methods used various biological properties to restrict the transition matrices of these models. In this work, we will mainly compare against Signal P6, as it is the only model predicting all signal peptides considered in this work, and it was shown to achieve very good performance. Additionally, the train validation and test splits we use are identical to those used in the work developed by the Signal P6 authors. We also include a few other prediction methods, but the caveat here is that all these other methods were tested using their publicly available web servers. Since their training datasets might have contained similar sequences to those we use in our final test measures, these performance measures are likely overestimates. I will now move on to present the main results of our work. For signal peptide identification, we use the Matthews correlation coefficient, a popular performance measure for binary classification tasks. The coefficient takes into account true and false positives and negatives, and is therefore a good measure for imbalanced classes. We separately measure 2 tasks. MCC1 is the easier one, where we compute the MCC measure for each SB type, while only considering non-signal peptides as negative classes. MCC2 is the harder task, where we compute the MCC considering also other signal peptide types as negatives. On the right figure, our method is colored in dark blue, and it is the first method to the left, and our main competitor, Signal P6, comes right after it. Our method outperforms Signal P6 on many organism groups and SP types, with some exceptions such as the MCC1 of SecSPase2, but therefore it is competitive in terms of signal peptide identification. Moving on to the cleavage site prediction task. For it, we consider the F1 score, aggregating the precision and recall measures, such that comparison between methods can be done more easily. We consider 4 tolerance levels, where a cleavage site is considered correct if it's predicted within various thresholds T from the annotation. The main reason for this is that CS annotations can be slightly erroneous. We outperform the previous state-of-the-art Signal P6 across most signal peptide types and organism groups, and almost all tolerance levels. Note that the CS F1 scores are negatively influenced by predicting an incorrect signal peptide type, and we therefore did not include other models here, as they do not predict all signal peptide types considered in this work. In addition to the homology split test results, we test our model on 4 novel eukaryotic sequences. These sequences are unusual in the sense that the NH and C subregions of the signal peptides are not clearly delimited. For example, they have no clear contiguous basic regions, colored in dark yellow, followed by a hydrophobic subregion, which would be colored in blue. These 4 sequences were identified by luciferase secretion assays, and our method was able to identify 2 out of 4 of these sequences as containing signal peptides, whereas no other previous method was able to detect any of them. Here, since we do not encode explicitly any biological information, we attempted to determine how well the model can distinguish various properties of signal peptides. We use the amplitude of the gradients of the model's prediction with respect to the inputs and determine the relative importance of specific regions of the sequences. These plots on the right show importance score specifically for test sequences. And here we have tested the twin arginine translocation motif importance when the model predicts a twin arginine translocated signal peptide. And we can see that the relative importance of the TAT motif is quite high. Also, the model places a considerably higher importance on the cysteine residue when predicting the cleavage site of SecSPase2 cleaved proteins. To conclude, we have developed the first fully data-driven signal peptide prediction method. We achieve competitive signal peptide identification and state-of-the-art cleavage site prediction performance. We identify prior biological information. And we have highlighted the importance of this approach on unusual signal peptide sequences. Thank you for your attention.